Hello students, my name is Gemini. Today we are going to start the first chapter of class 10 ICSE biology, that is cell cycle, cell division and structure of chromosomes. Before going to the cell cycle, just we'll discuss a brief about cell. As you already know, cell is the basic unit of all living organisms, and every organism, whether it may be a higher organism or a lower organism, every individual body is made up of cells. Cell was discovered by Robert Hooke in 1665, and there are different types of cells like prokaryotic cells, eukaryotic cells, and there are different number of cells in different organisms. So the number of cells varies from species to species. And how the cell is produced? Cells are produced from pre-existing cells. Next we will start with the cell cycle. As the name indicates, cycle, it is an orderly sequence of events. As you can see here, orderly sequence of events by which a cell duplicates its genome. Duplicate means producing a copy of its genome. Genome is the sequence of genes. Genes are the units of heredity which are located on the chromosomes. And as you all know, chromosomes are the hereditary material or genetic material present in the nucleus of a cell. Genome means the set of genes. Next, coming to cell division. It is the most important part of this topic, that is cell division. What is the need of cell division and why does the cell divide? In some lower organisms, cell division itself is a mode of reproduction, as in amoeba. There, cell division itself is a mode of reproduction. It is a process by which one cell produces two or more daughter cells. Two daughter cells are produced as in case of mitosis, and four daughter cells are produced in case of meiosis. That will come later. Next, coming to generation time. The duration between two successive cell divisions is called generation time. Once again, generation time is defined as the duration between two successive cell divisions. For example, in humans, the duration of cell cycle is about 24 hours. The duration is 24 hours. Every 24 hours, a cell divides. Next, coming to phases of cell cycle. There are two main phases of cell cycle. Number one is interphase. It is also called as non-dividing phase. Interphase. Number two, M phase. Mitotic phase, or also called as dividing phase. First we will discuss about interphase. Earlier, interphase was called to be as a resting stage, but it is not such. Actually, interphase is the most active phase where the cell prepares itself for division. In interphase, cell prepares itself for division because the next phase is the dividing phase. Interphase is further subdivided into three stages. First one, G1 phase, or it is also called as post-mitotic phase. G1 phase, or the first growth phase. Number two, S phase. S phase means synthesis phase. Here, synthesis of DNA takes place and the chromosomes get duplicated. Number three is G2 phase, or pre-mitotic phase, or it is also called as second growth phase. So once again, interphase is divided into three sub-stages, G1, S and G2. It is the most important question will be asked from this topic. Write the sequence of cell cycle, that is G1, S, G2 and then comes the M phase. So in these three phases, the cell is preparing itself for division, getting ready for division, and that is the next phase called as M phase. Now next we will see what happens in G1 phase. It is also called as post-mitotic phase. In growth 1 phase, protein synthesis occurs. Proteins, as you know, proteins are basically enzymes which are required for DNA replication. Here protein synthesis takes place, RNA synthesis takes place, and for all these synthesis, ATP is also required. So ATP or synthesis also increases. And in this G1 phase, the cell is preparing for the division, for the S phase. So in G1 phase what happens? Synthesis of RNA, synthesis of proteins which are required for DNA synthesis. Next, coming to S phase. S phase, here S stands for synthesis. Synthesis of DNA takes place. Now, what is the need for the synthesis of DNA? We take an example, humans. In humans we have 46 chromosomes. Now, if this cell divides, the two daughter cells also produces 46 chromosomes. Now, how it is possible? It is possible, now for example, let's take an example. Now I am having a page of information. Now if I have to give it to two students, I will take that page and give it to two students. But the information which is going to both the students is half the information. But if I want to give the complete information, then I have to take a photocopy of that paper. The same thing happens here. The copy of genetic material takes place so that the daughter cells are getting equal number of chromosomes. The same thing is happening in S phase, synthesis. DNA replicates, makes this copy and passing to the next generation. Next, coming to G2 phase or the gap 2 phase or the second growth phase. In this phase, the synthesis of DNA stops, but the RNA and protein synthesis continues for the mitotic division or for the next division as in M phase. So once again, G1, S and G2 phase, and G1 phase is the longest phase of interphase. Here the cell is most active, preparing itself for the DNA replication. In S phase, synthesis of DNA takes place, and in G2 phase, the synthesis of DNA stops, but the synthesis of RNA, protein, ATP continues because the cell has to enter the next phase called as M phase or dividing phase. As you can see here, the diagrammatic representation of cell cycle. It is in cyclic manner. It means that the events are continuously taking place. As you have seen the definition of cell cycle, the sequence of events are taking place in a cell to duplicate its genome. First, it is divided into two phases, interphase and the M phase. In interphase, G1 as we already studied now, G1 phase, S phase and G2 phase. So from here, this complete phase up till here, it is called interphase. You can see here, it is called interphase. And this remaining, this portion is called M phase or the dividing phase. It is called as M phase. And this M phase is further again divided into 4 stages, that is prophase, metaphase, anaphase and telophase. M phase consists of 4 stages, prophase, metaphase, anaphase and telophase. This will discuss a bit later, that is in M phase. So this is the diagrammatic representation of cell cycle. G1, S, G2 comes under interphase and the next is M phase or the dividing phase. So we already studied about the interphase. So interphase has 3 substages, G1, S and G2 phase. So in G1 phase we had seen protein synthesis, RNA synthesis, ATP synthesis and so many things are going on in the cell. What about the size of the cell? Size of the cell increases, so the growth has taken place, and mitochondria and chloroplasts also divide because they have their own DNA or the genetic material. Next we will come to the second phase, that is mitotic phase or the dividing phase. It is the dividing phase of the cell where the nucleus as well as the cytoplasm divides. Here the nucleus divides, that is called as karyokinesis. Karyon means nucleus and kinesis is something to split. So nucleus divides as well as cytoplasm divides into 2 daughter cells undergoing 4 important stages. So in mitotic phase, it is also called as dividing phase. Here the cell divides, that is the nucleus and cytoplasm divides into 2 daughter cells by undergoing 4 stages, important stages, that is prophase, metaphase, anaphase and telophase. These 4 are the stages of mitosis. So once again, mitotic phase is the dividing phase of the cell. Here the nucleus and cytoplasm divides. The division of cytoplasm is called cytokinesis. As I told you, cyto means cytoplasm, kinesis means to divide. By these 2 events, the cell undergoes division and produces 2 daughter cells, the prophase, metaphase, anaphase and telophase. Next, coming to types of cell division. There are 2 types of cell division. Number 1, mitosis, and number 2 is meiosis. First we will discuss about mitosis. What is mitosis? What is the definition of mitosis? Mitosis is a type of cell division where chromosomes are equally distributed to the daughter cell. Equally distributed as in parent cell. Because of this reason, mitosis is also called as equational division. There is another term for mitosis. Equational division. The name itself indicates equational means the number of chromosomes in parent cell is equally distributed to the daughter cells. Because of that reason, it is also called as equational division. And this mitosis takes place only in somatic cells. Somatic cells. So the division is also called as somatic cell division. It is also called as somatic cell division. Mitosis was first discovered by Strasburger in 1875 in plant cells and Walter Fleming in 1879 in animal cells. And it was named as mitosis in 1882. Once again, mitosis is also termed as equational division because the number of chromosomes are equally distributed to the daughter cells as in parent cell. So it is called equational division. And why it is called somatic cell division? Because it occurs in somatic cells. Somatic cells growth takes place. Next, it was coined by Strasburger in plant cell and discovered by Walter Fleming in animal cells. Now mitosis. Again, mitosis undergoes 2 stages. As I told you earlier, here we are mentioning a very brief here in mitotic phase, but here you are going to study in detail. Mitosis is further divided into 2 stages, karyokinesis and cytokinesis. Karyokinesis, karyon means nucleus. The division of nucleus takes place under 4 stages, prophase, metaphase, anaphase and telophase. After karyokinesis, the next stage is cytokinesis. The question may be asked as, name the stages of mitosis. Prophase, metaphase, anaphase and telophase. Prophase, metaphase, anaphase and telophase. And the question also may be asked as, why it is called equational division? It is a very important question because here the chromosomes are equally distributed to the daughter cells. And it is also called as somatic cell division. So in mitosis we had seen the 4 stages, prophase, metaphase, anaphase and telophase. After telophase, the new cells are produced and hence the cycle continues. As we had discussed earlier, cell cycle is a sequence of events, means all the stages are again repeating in the cell once in 24 hours. But this cell cycle is not continuous in all the cells. For example, in brain and nerve cells, in the embryonic stage, the cell division or cell cycle stops. So for example, in mammalian skin cells, as and then the new cells are produced every 24 hours. And a very good example is of RBC, red blood corpuscles. You know, the lifespan of RBC is 120 days, after which they are again replaced. So these are all some of the examples of the cell division. So till now we have discussed about the M phase. Next we'll continue with karyokinesis. Karyon means nucleus and kinesis is to split. So in karyokinesis is the division of nucleus. The karyokinesis is completed in 4 phases: prophase, metaphase, anaphase and telophase. Once again, karyokinesis is the division of nucleus. Here nuclear division takes place by undergoing 4 substages: prophase, metaphase, anaphase and telophase. Before going to the prophase, we have drawn the diagram of interphase stage first. As you already know, interphase is the preparatory phase where the cell prepares itself for division. Here we have taken the example of animal cell. Here we need not draw all the parts of a cell because the division takes place only in the nucleus. We have seen here nuclear pore, nuclear membrane. We have drawn a double membrane because you all know nucleus is double membrane structure. The space between this membrane is called as nuclear pore, and these thread-like structures, they are called as chromatin fibers. Now the chromosome number we have taken as 4. Just to keep a track of the number of chromosomes, I have taken the diploid number of chromosomes, that is 4. So I have drawn here 4 chromatin fibers. Here chromatin fibers are thread-like structures. Now we will see what happens in prophase. So there are so many changes taking place in the prophase stage. So we have divided it into early prophase and late prophase. First we will see what happens in early prophase. In early prophase, as you can see here, the nuclear membrane starts to dissociate and it disappears. The chromatin fibers start to condense. Again the number of chromosome is same here, 4. The chromatin fibers start to condense. The Golgi apparatus, nuclear membrane, endoplasmic reticulum, they start to dissociate and they disappear and chromosomes condense. And there is centrioles here, this formation of centrioles. As you already know, in the S phase, in the synthesis of DNA, centrioles also gets duplicated. Next, what happens in late prophase? In late prophase, there is formation of spindle fibers. From where the spindle fibers are formed? They are formed from these asters. These are called as asters. These small rays are called as asters, which give rise to spindle fibers. And here chromosomes are attached to this spindle fiber with the help of kinetophore. This is again 4 chromosomes here. So this is the prophase, early prophase and late prophase. So what happens in prophase stage? The chromosomes or the chromatin fibers start to condense. The nuclear membrane, Golgi membrane all dissociates, and next one is metaphase. In metaphase, chromosomes align at the equatorial plane. Now see the position of chromosome here and the position of chromosome here. Now they align on the equatorial plane. How they are coming to the equatorial plane? Because of this spindle fiber. There is contraction of spindle fiber here, which brings these chromosomes on this equatorial plane. Chromosomes are aligning on equatorial plane. Next is anaphase. Third stage is anaphase. In anaphase, chromosomes move to the opposite poles of the cell. Chromosomes move to opposite poles of the cell because of this constriction. And here there are 2 types of fibers: continuous fibers and discontinuous fibers. And in between these 2 fibers there is some space called as interzonal fibers. Now here the division is of 2 types: amphiastral and anastral. So in metaphase, here there is formation of centrioles here. 2 centrioles are formed here and they are moving to opposite poles. 2 centrioles. So the formation of 2 centrioles in animal cells, 2 centrioles are formed. So in case of animal cells, the division is said to be amphiastral. Amphi means 2. Amphiastral in case of animal cells. Once again I repeat, in animal cell there is formation of 2 centrioles and they are moving to opposite poles. Such kind of division is called as amphiastral. In case of plant cells, as there is no formation of centrioles, so the division is said to be anastral division. Anastral in case of plant cell. Amphiastral in case of animals, as there is formation of 2 centrioles, and in case of plant cells there is no astral formation because there is no formation of centrioles. So there is anastral division. In anaphase, the chromosome move to opposite poles of the cell, like a string. If you take a string and stretch it, it moves to opposite pole. So likewise, the chromosomes are moving to opposite poles of the cell. Next, coming to the fourth phase, last phase, that is telophase. In telophase is nothing but the complete reverse of this prophase. What had happened in the prophase and what is happening in the telophase? In prophase, we had seen the nuclear membrane all dissociates. Chromatin fiber condense to form chromosomes. But here, again the chromosomes condense to form chromatin fibers. Nuclear membrane reappears. So it is exactly the reverse or opposite of prophase. And here there is formation of 2 daughter cells. So this is about telophase. So this is about karyokinesis. Once again, prophase, metaphase, anaphase and telophase. We have drawn the stage of interphase to show how the division takes place. Prophase is divided into early prophase and late prophase. As there are so many changes taking place, so we have divided into early prophase and late prophase. Next, coming to metaphase. Most important thing you have to remember here is, in metaphase, chromosome align along the equatorial plate or equatorial plane. Anaphase. In anaphase, chromosome move to the opposite poles of the cell. And you remember these 2 points, amphiastral and anastral division. This is a very important point. What is amphiastral division? Amphiastral division means there is formation of 2 centrioles. Amphi means 2 centrioles in case of animal cell. What is anastral? Anastral division means there is no formation of centrioles. Example, plant cell. Next, telophase. In telophase there is formation of 2 daughter cells and the chromatin fibers are formed again. Nuclear membrane is again reappearing. So it is nothing but the entire reverse process of prophase. Next, coming to cytokinesis. As there are 2 steps, karyokinesis and cytokinesis. This entire thing is called as karyokinesis. In all these stages, division of nucleus has taken place. Nucleus has undergone division, so we call it as karyokinesis. Cytokinesis means division of cytoplasm. Cyto is cytoplasm, kinesis again to divide or to split. Cytokinesis. In animal cell, cytokinesis takes place by furrow formation or constriction. So here slowly there is forming, as you can see here, slowly there is constriction, and these constrictions come close together like this, and the entire cell divides into 2 daughter cells. This is the constriction. This comes close together slowly and there is formation of 2 daughter cells. But in plant cells there is no constriction. In plant cells, cytokinesis takes place by cell plate formation. In plant cells, cytokinesis takes place by cell plate formation. The most important thing you have to remember, in animal cell, cytokinesis takes place by constriction or a deep furrow. Slowly the cell, there is a deep formation like this and then they divide. But in plant cell, there is cell plate formation by which the cytokinesis process is completed. So, already studied the stages of mitosis. Now let's see the significance of mitosis. What is the importance of mitosis? As you already know, mitosis is called as equational division. We already studied that, equational division, where the chromosome numbers are, or chromosome number is equally distributed to the daughter cells. It is also known as somatic cell division because mitosis takes place in somatic cells. So let us see some of the significance or importance of mitosis. Number 1 is maintenance of chromosome number or genetic stability. As I already told you, maintenance of chromosome number. So the chromosome number is equally distributed to daughter cell. Same number of chromosomes. So it is called as maintenance of chromosome number or genetic stability. Number 2, maintenance of cell size. Mitosis helps in maintaining the size of the cell, or you can say mitosis helps in keeping the size of the cell constant. Number 3, growth. This is the most important significant of mitosis. If you take any organism, whether a plant or animal or a tree or human beings. Every organism starts its life with a single cell called as zygote. Zygote. After fertilization, fertilization, it is the process of fusion of male and female gamete. After the fusion of gametes, the resultant cell will be a diploid zygote. So zygote undergoes mitotic division to produce the new organism. Replacement, as the name indicates, replacement. So in our body, billions of cells die and again new cells are formed. That is called as replacement. When and then the cells are being replaced in our body due to the mitotic divisions. Next, repair. As the name indicates, repair. Sometimes we have some injuries or cuts or some surgical operations due to which our tissues are being worn out. That is also again it is repaired by mitotic divisions in our body. Next, asexual reproduction. What is the meaning of reproduction? It is the biological process where organisms give birth to young ones. There are two types of reproduction, sexual and asexual reproduction. Now here we will discuss about asexual reproduction. Asexual reproduction is uniparental, means here the single organism produces the young ones. Here there is no formation and fusion of gametes. The single organism is producing the offsprings. For example, amoeba, yeast, etc. They reproduce by asexual reproduction. And in these unicellular organisms, cell division itself is a mode of reproduction. So here also mitosis takes place, the cell divides and the new generation is produced. Next, coming to regeneration. Again the name indicates regeneration, means now we all know that about lizard, right? Lizard is the well known example for regeneration. Due to self-defense or due to some many other reasons, the tail has been cut in the lizard. Then that tail grows within few days due to mitosis, the cell division called as mitosis. The tail part is again growing into a new individual by the help of cell division called as mitosis. Next, embryonic development. In embryonic development, as I told you, after fertilization, there is formation of diploid cell called as zygote. Zygote further develops into embryo. How it is developing? By a division called as mitosis. So mitosis also helps in embryonic development of an individual. So this is the significance of mitosis. Maintenance of chromosome number, growth, repair, replacement, asexual reproduction, regeneration and embryonic development. So these are the significance of mitosis.